Okay, all right. Hi, Claudia. So, welcome to the IES Microplastic lecture series. So, our today's speaker is Dr. Claudia Halsband. She is a senior researcher with, sorry, it's difficult for me to pronounce. It's
Akvaplan-niva.
Tromsø from Norway. Her topic is microplastic pollution in the Nordic marine environment. Professor Dr. Claudia Halsband is head of the section ecosystem at the private research institute and the consultancy Akvaplan-niva with headquarters in Tromsø, Norway. In her research, Dr. Claudia studies anthropogenic impacts of climate warming, ocean acidification, and microplastics pollution in marine ecosystems, with a recent focus on the combined effect of multiple stressors on marine and Arctic ecosystems. Claudia is a member of the steering committee of the Fram Centre program, CLEAN. She holds a joint European PhD from University of Paris and Oldenburg. And she's also the former EU Marie-Curie postdoctoral fellow with research experience from posts in Germany, France, US, UK, and Norway. Thank you. So you can start.
Yes, thank you very much. And thank you very much again for inviting me to be part of this lecture series and letting me set the spotlight on the High North today. So I will talk about the European sector of the Arctic, and we'll try to share my screen now. Let's see.
Yeah, no problem. We can see it. Yeah,
excellent. So yeah, I'm based in northern Norway in Tromsø, so quite far above the Arctic Circle, and so this is going to be the focus of this presentation. So the more recent microplastics research has had an early focus on the marine environment, because this is where the early works from 2004 onwards have focused. So a lot of the plastic degradation and the plastic pollution, of course, in the end ends up in the global oceans. But more recently, we also have more information about freshwater and terrestrial systems, where, of course, the plastic pollution originates and where it comes from. So we need to connect these two realms of the environment. Now, when we think about microplastics, is of course a very human-centered perspective. We are at the source of this type of pollution. So we think of highly populated areas around the globe, and garbage that goes the wrong way, that degrades in the environment, and highly polluted plastic contaminated waterways. When we think about the Arctic or the High North, we usually think of this as a very remote and pristine environment. So we usually think of environments like this one. It's very popular with the tourists who come here and actually forget about plastic pollution and see a very clean and nice environment that seems untouched. But of course, microplastics are so small that they are invisible to the naked eye and also require quite sophisticated methods for extraction and enumeration, while in turn the Arctic is very remote, it's ice covered, it's difficult to get to, it's dark in the winter and difficult to work in those conditions, so we have a lot fewer samples usually from that environment than from other places around the world. So nonetheless, about 10 years ago, the first results emerged that even Arctic sea ice is contaminated with microplastics. So you see here a study from 2014 where researchers have found quite a diversity and quite high numbers of microplastic particles in Arctic sea ice from different locations around the Arctic Ocean. And as you see here on the graph on the left, that even in the middle of the Arctic Ocean, so right in the center, that should be the most untouched, presumably, you have the highest concentrations even. So that was a bit of a shock and a surprise that this is really also a contaminated area, that we find all sorts of different plastic polymers, so probably different origins that they come from, and that there are so many of them. And a year later, we also found that surface and subsurface waters in the Arctic are contaminated with microplastics. The methods are quite different, how we collect these microplastics. So it's usually the traditional methods that we use in oceanography, such as plankton nets, for example. And so you see very different numbers, but also quite a variety of different microplastics, particles, fragments, fibers that we see between mainland Norway and the archipelago of Svalbard, which is the area that you see here on the map. So we have microplastics in sea ice, we have microplastics in the water column, and sure enough, we also find microplastics at the sea floor in the sediments. So here's another study, again a few years later, that demonstrates that, again, we have a very high diversity of different polymers and different types of microplastics in Arctic deep sediments, in the Fram Strait in this case. So what is the deal with these microplastics? How do they get there? Why are they there? And how do they move around? So people ask themselves, why do we have these microplastics there, and where do they come from, and where do they go? And you see here some trajectories that have been calculated, and also some differences in the distribution of microplastics in sea ice that point to the fact that probably they have different sources, and that they are not static in the Arctic environment, but that they get transported around and moved around, that they get entrapped in the sea ice over time, that they potentially get stored there for a period of time before they also can get released again when the ice melts. And of course, with climate change, we have a lot of sea ice melting going on, such that the sea ice is not just a sink or a final storage place for microplastics, but at the same time also a source. So these microplastics can be transported back to our latitudes, to the Fram Strait and the North Atlantic that way. And so here is an example where people try to understand how these dynamics work and how mobile and dynamic the microplastics transport is. Then one thing that was striking in these studies that came out is that the numbers are quite different. So when you compare two of these studies that I presented in terms of how many particles of microplastics you find in sea ice versus the surface water under the ice, it looks like there's a lot more particles in sea ice than in the water, which again supports the hypothesis that the sea ice is a sink and that it gets stored there for longer periods of time, which might be part of the answer. But we also have to keep in mind that we are working with quite different methodologies here. Different studies include different types of microplastics and exclude others, depending on what they are confident their method can actually capture. So for example, when we talk about fibers, there's a lot of fibers that are not actually plastic, like cellulose or rayon are only either non-plastic or just semi-plastics. And so they are not always included in the studies. The same is true for all of the fibers. Some analytical methods have difficulties to actually enumerate fibers properly because they are long and skinny objects that are difficult to analyze. And so they exclude all the fibers. And again, you get different numbers if you exclude that big group, because a lot of studies find that fibers are actually the most numerous microplastics in some areas. So once you exclude these, you have a very different number than when you include them, and there might be a bias in your numbers. And finally, when we talk about microplastics, we are really talking about a a huge size range of particles. So depending on which size range this particular research group included, or which method allowed them to look at a certain range of sizes, again, you will get different numbers. So it will be very different if you look at particles smaller than 50 microns than if you're only considering particles that are larger than 100 microns, for example. And the plankton nets that I mentioned earlier, some only start at 300 microns to even capture these microplastics, because that's the mesh size of the net. So we have a difficulty comparing different studies, and are still working on, for this young research field, to find best standardized methods that everybody uses, and finding monitoring practices that will really demonstrate temporal and spatial trends of these microplastic particles in the marine environment. Now, one other important part that I wanted to point out about the sizes is again demonstrated here. So you see, this is an example again from the sediments in the Fram Strait, that we have all these different size classes. And what is important to note here is that the distribution across these different size classes is not uniform. So we have a lot more small particles than we have larger particles. And that makes a lot of sense, of course, when you think about that these microplastics originate from larger plastics items that disintegrate. And so one bigger particle disintegrates into many smaller particles. So it makes sense that the smaller ones are more numerous. But again, it also highlights the difficulty that particles below 100 microns, which are the most numerous ones, most methodologies don't even capture those, because that is still very difficult to go to such small sizes. And then we haven't even talked about nanoplastics yet. So that is a huge difficulty that is in the beginning of trying to be resolved, for example, by developing other sensors. So going away from the traditional oceanographic methods like nets, but develop sensors that use imaging, perhaps in combination with polymer IDing that you can do either in situ or from a sample that gets taken to the laboratory. But these methods are not currently available, and people try to develop those still. So we are still unknowns of how many plastics, microplastics are really out there. So, but when we come back to the question, is the Arctic or the High North a remote and pristine environment, we have to say goodbye to that idea. So the Arctic is highly connected, and you see here that the values are not all that much smaller than in the more populated and highly industrialized areas of the world. We also see that the types of plastic that we find in Arctic waters are quite comparable with other regions of the world. It looks like offshore or marine-based sources are a bit more represented than others. So, for example, fisheries seems to be quite a local source. So nets that are lost at sea are quite a high proportion and a little bit higher than in other regions. The reason for that the Arctic is not much less contaminated with microplastics than other reasons is, of course, the connectivity with the other parts of the world that you see demonstrated here. So we have both atmospheric transport all over the globe and also to the Arctic, and also ocean currents and aquatic transport that will bring some of these particles into the Arctic Ocean. Now, what is all the fuss with these microplastics? They are so small we can't see them, so are they really a problem, you might ask? So here's a picture with two different types of microplastic samples. On the left, you see these car tire rubber particles that are very recently a hot topic, and that I will talk more about in 3 weeks. And then there are fragments, there are these fibers. Fibers from textiles are a large fraction in a lot of areas where people took samples and say, these are the ones really to watch out for as the fibers. They're the most numerous ones. But they are really small. So here you see the scale bar of 1 millimeter. But the problem is, of course, that at the base of the marine food web, we have the plankton. And these organisms are in the same size range as the microplastics, and so is their food as well. Most of these little crustaceans that you see here that are called copepods, they will eat microalgae that you also see in the picture, these round and elongated objects that basically have the same size and shape as the microplastics we are talking about. So the risk that these two groups interact, that the zooplankton and planktonic organisms will interact and eat microplastics, is very high. So then, of course, the question arises, what kind of effects might these microplastics have in a Nordic marine food chain? And that is illustrated here. You have the microplastics that we measured in the sea ice, that we measured in the water column. So where do these get transported? Do they sink down in the water column towards the seafloor? Do they get eaten by organisms in the water column or at the seabed? Do these organisms that are in the size range of eating microplastics, do they get eaten by larger predators? And is there a transfer of microplastics from one group to the other? And does that in the end affect us humans because we like to eat seafood? So these are all questions that need addressing. And here you see the sort of the classical food web pyramid for the example of a high north ecosystem. And when you then look in the literature, then you will find out quite quickly that for most of the trophic levels that are involved here, microplastic contamination has been found, not only in lab studies, but even also in situ. So when you catch wild animals, they will have and show plastic contamination to some degree. So that's, of course, a cause of concern. And so the question then arises, what are the effects of all these microplastics that are out there that do get eaten or incorporated into organisms? Does that have effects on marine industries? And Norway has a very large aquaculture industry where these fish are basically in open fish pens mostly still today, although there's a lot of talk about more enclosed systems. But the usual way of aquaculture, and this is salmon farming in the case of Norway, these fish are just in the open fjords and open ocean and will be exposed to all the contaminants that are out there. The same is true, of course, for fisheries, where we just catch fish that has been exposed in the environment to whatever is out there. So there might be problems with these marine products that some economies are dependent upon. In turn, of course, these Nordic industries are also an emission source for microplastics potentially. And we have tried to summarize how much actually these, what we know about the emissions from these sources, but there are a lot of knowledge gaps. So we have illustrated here some of the sources one could think of, like, of course, the petroleum industry that has offshore installations, there's shipping, there is tourism, there's the aquaculture sector itself that, of course, uses plastic. Basically, everything has plastic items. As soon as there is an installation, it will have plastic involved somewhere. Now, a big new topic is offshore wind farming. So these installations have, for example, coatings against biofouling, but also are painted with polymer-based paints that can shed off. They are, of course, installed on the seafloor, or they float. So there are big ropes involved that are made of plastic usually. So there's a lot of different sources one can think of. I already mentioned lost fishing gear. That is a problem. So there's a lot of sources that are not really mapped very well at this stage, and where we need to know a lot more of what is out there and how it also can be prevented that these emissions occur. So we need to know more. How are we going to do that? So I think a combination of field sampling, experiments when we talk about impacts and effects, but also modeling studies are needed to scale this up to the ecosystem level and to larger regions. So I wanted to give an example here of how we can study microplastics distribution and transport. We tried to start small and do a little study in a small fjord in the Svalbard archipelago, north of the Norwegian mainland in Svalbard. and try to quantify. In this case, we wanted to focus on fibers that came from this settlement, so the small 2,000 inhabitant village in Longyearbyen in Svalbard, and they just, because it's a small settlement, they just pump their wastewater right into the fjord, and the idea is that it just dilutes in the big ocean, and that it's not an environmental problem as such. But we wanted to study the behavior of different types of plastic fibers in the system. So we had data, or we could enumerate the fibers in the actual wastewater. And then we wanted to know, as soon as they come out of the sewage pipe, how will they behave and be transported in this semi-enclosed fjord system. So it's a relatively small system that we can tackle a little bit easier than a whole ocean basin, for example. So we wanted to know how are they transported? Does the weight or the density of the polymer, there are of course all sorts of different polymers that have different properties. Do these properties have an influence on the transport behavior? And what does that mean for planktonic and benthic animals that might encounter these fibers? And what are the proportions of this? How many fibers versus how many organisms are we really talking about here? Well, here you see these fibers that we also use in experiments. So small, fluffy things, basically, from different types of plastic polymers. And we also included wool here, for example, which of course is not plastic, but is a good example for a very heavy, highly dense fiber. So for that purpose, it's a good example. And here's Akvaplan-niva in Svalbard that I was talking about. So where the red dot is on the map, this is where we are. And you see on this picture on the top there that a river comes in. So basically melting glacial water that comes into the fjord. This is why you see this brown plume on top. And the red dot with the arrow marks the outlet of the sewage pipe. And on the lower half of the picture, you see this wastewater hut where the wastewater pipes basically all combine from the village. You see the village in the background. And this is where we can actually go into this hut and just take a liter of wastewater out and actually count the fibers in the wastewater. So these are real numbers that we have determined in the lab. And then again, you see the wastewater pipe on the picture on the lower right. So they have already sort of taken precautions to make sure this wastewater doesn't just go and run out on the beach, but that this pipe is going into the deeper part of the fjord, quite far into the middle of the fjord, to enable dilution with the currents and the tides and make sure this water really gets processed by the natural system and cleaned that way. So here's what we found out. We have applied a model system, quite a high resolution model that we let run for 100 hours, so a relatively short period in time, just to keep control of what's going on. And we wanted to compare 3 different types of fibers, light fibers, neutral fibers that have the same density as the water, basically, and then heavy fibers that are heavier than seawater, and see if that makes a difference in combination with the hydrographic conditions and the oceanography that is going on in the fjord, so tides washing in and out, and the general topography that determines the current in that fjord. And let's see if this runs. So you see how these fibers come out and then distribute over these 100 hours in the fjord. The 3 pictures show 3 different depth levels. So you see on the left that it says layer number 1, that is the surface. The middle one says layer number 17, that is sort of mid-water column. So in the middle between the sea surface and the sea bottom. And then on the right, you see number 34, that's basically very close to the sea floor. And you see that these 3 different fibers have quite different behavior. So the light fibers get transported quite far out of the Akvaplan-niva into the larger fjord system on the outside. They seem to sort of go around the corner and then distribute, while the neutral fibers, you don't see all that much. There's a little bit of a shade, which means we didn't quite hit the depth layer where most of these end up. So they are not in this middle layer. They're either at the surface or at the seafloor, where we can't really see them very well in this simulation. And then you see on the right the heavy fibers that have a very different place where they end up in comparison to the light fibers. They stay in the fjord and actually get transported more towards the inner fjord. So we thought that was quite interesting. And here's another snapshot where we now try to analyze the data in a bit more detail. It shows again 4 different cases for different fibers. You see the letters deer here for Longyearbyen. This is where the settlement is. So this is where the houses are and where the wastewater comes from. And you see where these different fibers ended up after these 100 hours and where we have the highest concentrations. And that this was quite different for these 4 different cases. So the coast on the east, so the left side here, where most of the human population is actually located, was the least impacted in this particular example. Most of the fibers accumulated on the opposite, the west shore. And while the light fibers leave the fjord, the heavy fibers deposit within the fjord in the inner part of the fjord. So now, of course, this is a modeling study, and models are always wrong, as you might know. So we were lucky enough to have colleagues from the Norwegian Polar Institute that last year actually went out and took samples around Svalbard, including a few stations that are close to the places where we had our model run. And so I wanted to compare the real life samples with our model. And sure enough, they are quite different from each other. So the measured microplastics concentrations were a lot lower than the high peaks that we found with the model. Some of the trends are similar. So if you look at the northern stations where we have this really very high peak from the model, but what you see is that it gets less towards the north, and that is the case also for the measured particles. So some similarities, at least in sort of the trends, are there, but there's also the 2 stations in the south where it's basically the opposite, where we find no contamination and they find higher values than at the other stations where the model predicts high concentrations. So this is just an eye-opener to not believe anything the model says necessarily. We are trying to learn with these models to predict and understand the system. But ground truthing is a necessity here to really make sure that the trends and the patterns that we see, that the model is actually capturing them. Now, another thing to remember here is, of course, the temporal dimensions. We were simulating something over 100 hours, while what you measure there with a plankton net or with water samples at any point in time is a snapshot of integrated concentrations over a time period that we don't really know what it is. So it's very difficult to compare these 2 things. And it would be nice to compare more of these samples and more model runs to see if we can just understand the principle of this transport a bit better. Now, what we were also interested in with this modeling study and the samples is what happens when microplastics come across organisms, or the other way around, when the organisms come across these microplastics. So what could be the impacts when organisms meet microplastics? So as I said, they normally eat diatoms and other microalgae that are in the same size range as the particles. So what we tried here is with the same fjord and the same data to estimate how many organisms will overlap in time and space with the fibers that we were looking at. So we have the light, the neutral, and the heavy fibers, and we basically calculated this together with abundances of plankton that we had for this area. And you see that, of course, pelagic organisms that are also quite abundant in the surface area are most impacted or will have most encounter risk at this shore where the model predicts the high surface concentrations, and a bit less for the heavy fibers because those are quite deep in the water column. The opposite is true for the benthos, so the seafloor organisms. They are almost not at all impacted or have no overlap with the light fibers. They have mostly overlap in the inner fjord with the heavier fibers. So the impact or the ingestion rates of fibers of these different properties will be very different for different functional groups of organisms. That is basically the take-home message here. Now, to sum this up a little bit, so both plankton and benthos have overlap with microplastics in space and time. So the risk of encounter is quite high. It's driven by the oceanography. So currents and tides have to be taken into consideration and the dynamics of those. The properties of the microplastics themselves play a role for the transport. Smoothing approaches to match the modeling results with the actual samples. Both seem to be wrong in some way. And then we have seen that a large group of different organisms will have some sort of ingestion of microplastic types across all the trophic level that you find in the food chain. So what are the reasons for these interactions between microplastics and organisms? Or you might just simply say, why are organisms so stupid to actually eat plastic that has no nutritional value? So there's a number of reasons. First of all, there are, for example, planktonic organisms that just filter water, and they are quite unselective. So they have no mechanism to avoid incorporating or ingesting anthropogenic particles. That's just the way they are built, basically, because normally in the marine environment, you would not come across any particles that are unwanted. So the biology determines that they just filter out particles no matter what they are, and that they are normally organic particles. Then, of course, microplastics can attach themselves to structures, to the algae, for example, to marine snow, or to the body structures of a prey item. So then it's sort of accidental ingestion of the microplastic. Another interesting aspect that we found is that the plastic doesn't just stay clean plastic, but there's biofouling that goes on, or the plastic gets incorporated into microplastic marine snow aggregates that then basically have a nutritious disguise for predators or for feeding organisms that will consider this as food. A lot of microbes also emit infochemicals. They have, in a way, a smell for the predator, so they will attract ingestion that way. And then, of course, once a particle gets eaten by one thing, and that is the prey of something else, there might be this trophic transfer from the prey to a predator that we call biomagnification, so that a predator would accumulate a lot of microplastics by just eating its regular prey that in turn is contaminated with the microplastics from feeding. So we wanted to know a bit more about this ingestion mechanism, especially for these planktonic organisms. So we have compared 4 different species here, and basically, with the exception of one, the Pseudocalanus here in the middle, all of them ingested microplastic particles, some more, some less. But the really interesting part was to compare these clean particles with biofouled particles. So particles that we have aged in natural seawater for several weeks, so that they had this corona or this biofilm on them. And that was, we could statistically see that this fouling caused more of the copepods to actually ingest these particles. So they are a lot more attractive once they have this microbial biofilm on them. And that causes more ingestion than just a clean particle. Yes, so to sum up a little bit, what do we still need to understand Nordic marine plastics impact? We have tried to summarize this a little bit especially for the Norwegian case, where we are quite in a good position, where we know a lot of things already and have also mapped the knowledge gaps of how we could tackle this, how we could integrate research and monitoring what we need to know about the sources and emissions, discharges of microplastics, how we need to understand better the mechanisms that make macroplastics, so the items that we throw away, how they become microplastics. This is actually something where we are concerned about so-called biodegradable plastics that have been discussed and are sometimes used for plastic bags, for example, which are not bio, but they just promote the degradation. So you just make microplastics faster than with regular plastic, which is not really what we want based on everything that you have just seen. So that's not a solution in our eyes. So biodegradable should really be an organic material that has no plastic whatsoever involved. So these are all discussions at the societal level that we need to have. Then, as I try to demonstrate, fate and transport of plastic pollution is still not understood in enough detail. The consequences of uptake and ingestion, how that translate into effects is very sort of diverse effects. You see negative effects, you see no effects. This is again, there's a lot of wealth of data now for different organisms, ingestion and also egestion and trophic transfer, but we don't really have a good handle on the effects quite yet to translate this into a risk assessment. And that then needs to trigger public discourse and regulations that will reduce the emissions and the impacts of microplastics. So we try to outline this and have a little suggestion of how to implement this. And most recently, we were also discussing in a Nordic consortium what the next questions are that the research should resolve. So monitoring and the regulation is a parallel process, but there are also these knowledge gaps that we still need more basic research for. So here again, you see basically the Nordic environment with a highly connected different oceanic regions. That includes also the Baltic Sea and the North Sea in the south, but the Barents Sea and the Arctic Ocean in the north, and of course areas in the North Atlantic and around Iceland as well. So we formulated a number of research questions that you see here, and welcome to read the paper, that outline the knowledge gaps that we see as most pressing and also most interesting. And that is a whole range of different questions that have to do, again, with transport, also long-range transport from the industrialized areas into these more remote areas and how this works, both on the atmospheric level and with the ocean currents. Also to combine the monitoring and understanding of regional transport patterns. We need to know more about additives in plastic and how those behave, because there's of course just the plastic polymer that is relatively inert, but most plastics do not just come as pure plastic material, but they have a lot of additives, and those additives can be even more problematic than the particles themselves. And there are combined particle additive effects where not just the chemistry of the additive is problematic, but in conjunction with the particle that is administered, it might have a different effect than just the chemical alone. We need more model simulations to visualize transport and also ecosystem effects and upscale to the ecosystem level. And we need to understand better the spatio-temporal patterns, because of course we always look at sort of static two-dimensional pictures, but especially in the marine environment, everything is very dynamic, everything changes over time. So just looking at these snapshots, a sample that I take on one day is not going to be representative of the next day and not of a place 10 kilometers down the line. So the whole dynamic of the problem is something that we still need to understand better. And then, of course, finally, if there is ingestion and trophic transfer and incorporating also the smallest particles, I've mentioned nanoplastics very briefly and that they are not well mapped, but we know from laboratory experiments that those are the most problematic because they really can go through membrane barriers. They can be found in brain. They go from the digestive system into other organs of organisms, and this is where we think the impacts will be, rather than just ingesting larger 100-micron particles and ingesting them again. But the really small ones are the ones that will cause the trouble. So immunotoxicology is a keyword here, and also the toxicity of particles in combination with additives. Those are the big questions. Yes, so to sum up a little bit, the conclusions here. So high north marine systems, unfortunately, are contaminated with microplastics, and that applies for all the environmental compartments from ice, water, sediment, and biota. We have knowledge gaps and need still standardization to be able to compare across different studies. The first monitoring efforts have started, but as I said, they don't really target the smallest particles. Microplastics need to be considered in a three-dimensional system plus time, so the temporal dimension needs to be incorporated as well to understand distribution and transport. Yeah, as I said, temporal patterns are understudied at the moment, and it's difficult to go out over and over again with an icebreaker and take real-life samples. So that is one of the obstacles, where then the modeling needs to be improved with these real-life samples. Then we've seen that microplastics interact with organisms. Some just get ingested and egested again, but as I said, the smaller ones might be more problematic. Bioaccumulation and trophic transfer in food webs are possible and need to be better studied. The smallest one poses the highest biological threats, and the additives need to be put into the mix here to assess the environmental risks. Yes, and with this, I want to acknowledge some of my colleagues that have worked with me on all these different questions, and 2 of the projects that have contributed to this. And then I'm happy to answer questions.
Okay, thank you. Thank you, Claudia. So now it's question time. Any questions? I think some people leave a message here. So the question is that what are the major challenges in monitoring and assessing the presence of microplastic in such pristine remote areas? Is it possible to come up with a probe to identify microplastics while sampling?
Yes, that would be very nice. And that is the dream that we have right now, that we can use remote vehicles that don't even require us to be there in person, that can ideally go under the ice and be remote control from the office, and that will collect this information. We do have the remote vehicles, and they collect all sorts of information, but we are not quite there yet for the microplastics probe. But I've just been contacted by people in Trondheim, also here in Norway, that are thinking of this idea and that were interested in the remote vehicles and that are trying to develop a Raman-based approach to deploy in situ and to sort of sniff up microplastics right there and then. So it might be down the line that we can develop something like this, but it's really still early stages to develop a method that can then actually be deployed and maybe bought off the shelf and used for monitoring in a lot of areas.
Thank you. Thank you.
Can I ask a question, Claudia? Your comparison with your modeling and the actual sampling that took place in the fjord, you mentioned that the time base is quite different, but I'm wondering if part of that difference, as you were looking at the very light particles, is just obviously they're going to be moving much faster. And so I guess my question is, would you then expect for the light particles that the distribution would be fairly uniform if you started to do a more widespread sampling across like the entire Arctic or even out into the open Atlantic?
Yes, so that would be my expectation, that they will be quite widespread and also more uniformly distributed in the end, because of course then we are looking at a long-term average, right? While this is just sort of releasing a plume once and then following it. while any sample that you will take now will integrate the contamination from the last 50, 100 years across the entire area. So yeah, that needs to be taken into consideration. And yeah, I would agree that probably the lightest ones are the most uniformly distributed.
Can I jump in with a quick question as well?
Please.
Just to follow up from Doug and Claudia, great talk. Very much enjoyed it, but I'm just at home and there's other noise and stuff here, so apologies. I'll just try and sneak in a question. You mentioned biofouling, and I did wonder in terms of the model simulation, if there is such a thing as a light fiber from a sewage treatment plant, or do they all become biofouled and encoded and more or less act like heavy fibers? So I did wonder if you looked at the sediment in and around the fjord as well, and if you did find them dominated by heavy versus light fibers.
Right. No, unfortunately we don't have sediment samples yet. It's again one of the plans to look at those as well. We do know that most microplastics and fibers do get biofouled quite quickly when they are in a natural environment that has a lot of microbes. So most of them will do that, and yes, it will impact on sort of the heaviness, or the light fibers will get heavier as they are biofouled. So that is another thing that the model has not incorporated in that case, but that will change the properties over time of that particular fiber or microplastic. So yeah, that's another complication.
Yeah, it's always complications, and again, some of those with the biofouling, they may even flocculate together, forming clumps.
Yes.
But yeah, I think it would be good to carry out some sediment sampling in the fjord. If you need any field hands, don't hesitate to ask.
Yes.
And I look forward to those results. Thank you.
Excellent. Thank you.
Another question from the audience. So the question is that, are microplastic in Nordic marine environment tend to mobile or immobile or accumulate? What's the main factor?
Yeah, that's a good question. It's difficult to answer without more samples, but I think they will accumulate eventually. We have debated that back and forth also, where we should take the samples to take a shortcut and go away from the surface sampling, because eventually with the biofouling and other mechanisms, everything will rain down eventually to the sediment. So there is still debate what the best place is to look. The sediment certainly is one place, but there's also some modeling studies, and again, they are modeling studies, but that say they will just be washed around the global ocean basins endlessly, basically. So there will always be some that will be resuspended or will be light enough to not actually reach the seafloor. So it remains difficult to just say, if we just look in one compartment, then we'll have the answer. So we need a more integrated approach, I think.
Okay, thank you. So I also have a question. When you talk about the aged microplastic, you said it's more like the more bacteria attached, right? The biofilm.
Right.
So I think we also found a similar result in our research. We found that after it's photo aged, so the microplastic is easy to be digested by some small animals.
Right. Yes. So it also changes the physical properties of the particle. So it gets more brittle, it gets more holes and sort of little structural. Yeah, the surface where also then the microbes can colonize more easily. So the age is again a complex sort of process over time that doesn't just involve the microbes, but also involves the physical and chemical properties of the particle.
Right.
Also where the additives then, some leach out at the surface, others stay still in, and so there are gradients of the chemicals, and that will have a distribution within the particles. So again, it's quite complicated. But yes, you are right that the properties of the particle itself also will change, not just the biofilm.
Thank you. Yeah. So any other questions? So if no questions, so that's another thank you, Dr. Claudia. Thank you very much.
Thank you very much, and see you in 3 weeks about car tire particles.
And the dog may say something, right?
Yes, thank you. Thank you, Claudia. Excellent talk. We do look forward to hearing you again, and as you say, in 3 weeks. If for those of you who have been following this series, you'll know that I did mention a few weeks ago that the IIES will be having its annual Science and Policy Workshop in Chile in December. And I just want to make one final announcement because we've reached the registration deadline. But if there's anyone who's listening who is interested in joining us in Chile for this meeting, you can find more information on our website. And we would take a few very late registrations. So if there's anybody, as I said, who is still interested in coming, please go to the website and register as soon as possible. So thank you. And thank you again, Claudia, for your talk.
Thank you.
Thank you. All right. Bye-bye. So we'll have next week the same time. So our series will continue. Okay. All right. Bye-bye. Good morning.